Now we're going to transition to, we're still going to be talking about proteins, but we're going to transition to Dr. Spangler. Let me give you a spotlight. Awesome. Okay, welcome Dr. Spangler. She is an assistant professor in biomedical engineering at Johns Hopkins University, and she's going to be joining us today to discuss her work with engineering proteins. Dr. Spangler earned a BS in biomedical engineering at JHU and went on to conduct her PhD research in biological engineering at MIT. So without further ado, don't want to spoil too much, so Dr. Spangler, please take it away.
Thank you so much, and thank you for this incredible opportunity to present to all of you today a little bit about my lab's research. I was really excited to see in the previous presentation some interest in, sort of the genomics and ultimately what goes into making proteins, and also how computational work can integrate with experimental work to create really cool science. And sort of continuing along that theme, I'm going to tell you a little bit about how my lab is using molecular engineering, specifically engineering things at the level of proteins to be able to modify the way that human physiology works. And particularly, we're very interested in how we can modify the immune response. And so without further ado, let me get into my slides. So many of you are probably familiar with the rapidly growing protein therapeutics industry, but interestingly, it's not actually that old. And in fact, not that much older than I am. And well, I guess I'm pretty old, but I'm not really that old. So the first protein therapeutic was actually first approved in 1982, which is, again, not really that long ago. We're talking about, 38 years ago. And I don't know if any of you can guess, but it was recombinant insulin. Which is, of course, what many diabetics take and are dependent on to replace that hormone that's lacking in their body. And in that 38-year span, right, from 1982 to today, there's been a huge explosion, right, of this market of protein-based therapeutics or molecular therapeutics, which are also called biologics. And there are over 300 biologics that are currently approved for use in humans, with a growth rate for biologics in the biologics sector of 3 times that of the overall pharmaceutical market. So as our population ages and various environmental and lifestyle factors come into play, there's an overall increase, right, in the demand for therapeutics. And to be growing at 3 times the rate of the entire pharmaceutical market is quite alarming. But when you think about a protein in nature, right, it doesn't care about your drug, right? The natural proteins are not pressured or evolved in any way to make good drugs. And in fact, there's a number of limitations, right, inherent properties of proteins that actually make them horrible drugs, right, terrible. And so the first limitation is what's known as pleiotropy. So when you're a protein, as we just heard in the last presentation, there's a lot of genetic information, right, DNA, that's encoding all of that information, or encoding everything that it takes to actually make that protein, right, and express that protein. And so that can be expensive. That takes a lot of energy, right, for all the processes of transcription, translation, and how do you, minimize the effort is by reducing the number of proteins that you have present in your body, right? So each protein is pressured by nature to be jack of all trades, right? Do a lot of different things at once in a very small genetic package. And that's great if you're a protein in nature, right? And trying to be very efficient, but that's terrible if you have a therapeutic, right? Because then it's going to be able to activate multiple different receptors on various cell types and have all kinds of effects all over the body, not necessarily, right, the effect that you would want. Let's say you have a drug that you want to target cancer, but if it's targeting all of your healthy cells all over the body as well, then it's not going to be able to perform its function for two reasons. First, it gets diverted, right? So the protein gets sponged up and soaked up, right, by all of these different healthy tissues as sort of a decoy, and it can't actually get to the location that it needs to treat the disease. In addition, by affecting those different healthy cells and having some sort of cytotoxic or cytolytic function, there's going to be a plethora of off-target effects, right? Meaning that it can have a lot of different side effects, which in many cases can be dangerous and lead to death of the host individual that's being given the drug from the therapeutic rather than from the disease that they have. The second limitation, which is common amongst protein therapeutics, is what I'm calling expression and stability issues, right? So in terms of recombinant expression of proteins, which my lab does a lot of, it's sort of the bane of our existence in many ways. Sometimes proteins express great and you're very lucky, but mostly proteins, right, again, they're evolved to be expressed in your body. And when you try to produce them outside of the body, right, some sort of exogenous way, you can get very low yields for production. They can be prone to aggregation or they can absorb to surfaces. And as a result, there are difficulties in manufacturability, and there can be a problem known as immunogenicity, right? So immunogenicity refers to an immune response against the particular drug that you're delivering rather than, again, against the disease, right? A lot of drugs are meant to activate the immune response, but activate that to kill a particular disease like a pathogen or cancer, not to actually activate the immune response against the drug itself, rejecting that drug and leading to potentially overactive immune and autoimmune pathogenesis. Finally, proteins have very poor what are known as pharmacokinetic properties, which refers to the rate at which they're cleared, essentially, what your body does to the drug and the rate at which they're cleared from the bloodstream. So when you administer a drug, typical protein of less than 50 kilodaltons, it will be cleared from your bloodstream in less than 5 minutes. And that's great if it's a diagnostic drug, right? If you're just trying to look at and localize that to a particular site of disease and then have it cleared. But that's terrible if you want to have a long-term sustained effect, right? In terms of a therapeutic. So again, that will lead to a transient response and inefficient delivery to the target organ. Okay, so I've stated all the problems, which is always, the first step, right? Figuring out what the problem is. But what's the solution, right? How do we solve this? And so my lab is very interested in how can we take what nature has given us and redesign, right? Rejigger those proteins, or inspired by nature, build entirely new proteins that nature's never even seen before that could be useful as therapeutics. So the way that my lab does that is we have sort of this three-pronged approach. And the first prong is what I'm referring to as molecular engineering. Now, molecular engineering is sort of a loaded term that can mean a lot of different things to a lot of different people. What my lab takes it to mean is using these processes of molecular evolution. So all of you, I'm sure, are familiar with, sort of the Darwinian concept of evolution, right? How we came from other forms of life and have evolved over time. So we can actually stimulate this process on a very small scale and on a very rapid scale in a test tube. And the way that we do that is using a display platform. So my lab uses the display platform of yeast, which a lot of you deal with on a daily basis in the bread that you eat. This is just simple baker's yeast. And what we do is we take these yeast and we zap them with different proteins, and we create what's called a library. Now, not a library where you read books, although I don't know that anybody uses libraries anymore in the digital age, but a library in the sense that it's a panel of different variants. So you start with a protein shown in red, and you make a bunch of different mutations shown in green and orange and cyan. And then the idea is that each one of those variants, right, is expressed uniquely on a different yeast cell. And then you come in with your bait, right? So when you go fishing, right, you want to have a bait to pull out the things that you want. And it's exactly like that. You go fishing in this library against this gray protein here, that's your target, and you pull out all the things that bind, you wash away all the ones that don't. And through these iterative cycles of directed evolution, right, which is, again, essentially a simulation of the process of evolution on the level of proteins, you can actually pull out the clones for the different sequences that represent those proteins that have the function that you want, right? So this is a really exciting concept, and it actually won the Nobel Prize a couple of years ago, back in 2018. But my lab obviously didn't discover that. We're not the first people to use this. So what's our unique angle, right? What do we do differently than people that are, it's doing straightforward or random protein evolution? So what we do is we try to integrate insights that we have from structural biology. So crystallography, cryo-EM, NMR, all of these different techniques and technologies that have been used to understand protein-protein interfaces have really catalyzed new opportunities in the field of molecular engineering, where we no longer have to just kind of randomly assign, right? How do you choose what goes in this library? Now we can actually build smarter libraries that can get us better, right, and faster to the solution for interesting proteins that we want to create. And the arrow goes both ways, in that once we actually discover these new proteins, right, that have the function that we want, we can solve their crystal structure and get at how they're working and how we can potentially make them better. And this is great, having this sort of symbiotic relationship with structure and molecular engineering, but we obviously want this to go somewhere, right? It's all about the translation, right? And about the next step. And particularly my lab, being at Johns Hopkins University, has a unique opportunity to integrate with clinicians and medical professionals who are able to bring these and translate these technologies into patients. And so my lab really works on spanning the space from the discovery of new molecules, designing and engineering them, and then ultimately being able to develop them in the preclinical and ultimately clinical settings. So in terms of their functional evaluation, we do that often in mice as sort of a simulated model for how the drug or therapeutic would act in a person. We also actually take our own blood and look at our immune cells and see how our different interventions and therapeutics impact those cells. And ultimately, the goal is to be able to translate these into phase 1 clinical trials. I think at a lot of institutions, you'd sort of say, okay, we can figure that out and, long-term kind of outsource. But what's fantastic, again, having the resources and opportunities at Johns Hopkins, is that we have the relevant patient populations as well as the infrastructure here to do that all in-house. And in the 3 years that I've been here at Johns Hopkins, I've actually been involved with 7 different patents of molecules, one of which we've licensed, and another of which we're building up in-house to develop into clinical studies, hopefully by the end of this calendar year. And so, again, I think the opportunities to really use these fundamental protein engineering technologies to take those all the way to the clinic is really what motivates a lot of the research that we do in my lab. And then the final arrow goes back, right? So once we actually, again, discover these new molecules and these new technologies, and we put them into people, we learn something, right, from the actual implementation in humans. And we can feed that information back into the process, right, the engineering process, to improve the therapeutics that we design. So what kind of drugs am I looking to, sort of create, and what sort of diseases am I going after? Well, as I mentioned at the beginning, we have a particular focus on immune diseases, and on a very high level, right, you can think of the immune system as a balance between immunostimulatory cells, things like CD8 positive T cells, NK cells, natural killer cells, other types of effector immune cells, saying, go, 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 kill those pathogens, right? Kill COVID-19, kill cancer, right? And there are certain diseases, as the ones that, for example, the ones I mentioned, where you would want to actually increase the number of effector cells, right? To again, bias the immune response to kill disease. Conversely, there's diseases where you would want to go in the other direction, right? Things like autoimmune disorders, diabetes, Crohn's disease, colitis, MS, right? Where your body is attacking your own healthy organs. And you would actually want to repress that pathogenic immune response. Also, if you're doing transplantation medicine, if I want to give somebody an organ transplant, I want to make sure they're immunosuppressed because I don't want their immune system rejecting the new organ that's going to save their life. So how do we use protein engineering, right, to create proteins that will bias the immune system, right? This is sort of our mission for targeted disease therapy. So then the next obvious question is, well, what diseases do you work on? And I often spin around the question and I say, well, what diseases don't we work on, right? Because when you're working at the protein level and at the level of, sort of molecular engineering, that applies to everything, right? The proteins are the building blocks of all forms of life. And so we actually work rather than, sort of taking a particular disease and saying we're a flu lab or we're a cancer lab or, we're a Crohn's disease lab, right? We actually turn where a lot of labs, have a particular disease target, and then they have a bunch of different approaches to sort of target that particular disease. We turn this paradigm on its head and say, we have this common approach of structure-based molecular engineering. And now we can apply this to a range of different diseases. And again, just to give you, sort of a high level overview of some of the projects we have ongoing in the lab, we have everything from activating the immune system in breast cancer, pancreatic duct adenocarcinoma, projects in HIV, malaria, flu. More recently, we have a project on COVID-19. And conversely, we have projects where we're trying to suppress the immune response. We have active grants in diabetes, skin transplantation, colitis, and also in tissue engineering, which has a lot of synergy with the immunoengineering field. So, I want to, in the last couple of minutes, just give you one quick overview of a project that will show you and illustrate for you how all of this works together, right? How does structure-based molecular engineering lead to therapeutic design and ultimately, disease therapy and immune disease therapy? So I'm going to talk a little bit about a project that we have ongoing where we're trying to engineer using actually computational protein design methods in combination with molecular evolution, new cytokines that will affect immunotherapy. So cytokines are soluble proteins of the immune system, and basically they're messengers that communicate information from the outside of the world into cells. And the way that they do that is by interacting with and dimerizing or binding together these transmembrane receptors. And the extracellular portions, as you can see, of these receptors coming together leads to their intracellular portions bumping into each other, setting off a cascade of events that ultimately will determine what the cell does, right? When you're a cell, you don't have that many choices in life, right? You're going to die, you're going to grow, you're going to move, you're going to, proliferate. But all of these different things that a cell can do and cell fate outcomes are controlled by cytokines, right, which communicate information into the cells at the protein level. A particular cytokine that we're very interested in is known as interleukin 2. And this was the first cytokine to be both genetically and structurally characterized. And it's an interesting cytokine in a lot of ways because it has two different types of receptors. So rather than just interacting with two transmembrane receptors, beta gamma, there's also this alpha receptor for cells that have express high levels of IL-2 or alpha. And importantly, cells that express this alpha receptor have a 100-fold higher affinity. So they normally IL-2 is biased towards signaling on cells that have IL-2 or alpha. And importantly, regulatory T cells express very high levels of IL-2 or alpha. So whereas effector T cells express very low levels. So normally IL-2 is biased toward creating an immunosuppressive environment, although it can also be used to create an immunostimulatory, right? Because it's sort of activating both sides of the coin. So even though IL-2 is normally biased toward Tregs, again, what if we want to stimulate the immune response against something like cancer, and we want to actually reverse that bias toward IL-2 or alpha low cells or effector cells? And so that's where we teamed up with David Baker's lab, which is located at University of Washington in Seattle, and is the premier lab in computational protein design that developed a software known as Rosetta that some of you may be familiar with. And this particular protein that we designed was meant to mimic a cytokine, specifically to mimic IL-2, but to improve upon the properties. So as you can see, the cytokine that we were basing this off of, IL-2, is a 4-helix bundle, meaning that it has 4 alpha helices connected by interhelical loops. And the interactions with the beta gamma subunit are, again, what would activate effector cells, whereas interactions with alpha would recruit in regulatory T cells. So to focus on effector cells, we focused on the interactions with the beta gamma receptors, and we fixed those in 3-dimensional space, and then built a cytokine, right, a globular protein around that, that basically integrated all of those different interactions with beta gamma, but no alpha. And another cool thing that we did was we said, in nature, there are 37 known discovered cytokines, and there could be more, but they all have the same format, that their helices are facing up, up, down, down, meaning that the first 2 end to C terminus are going up and the second 2 are going down. And that's actually very inefficient, because to get from the top of the first all the way to the bottom of the second, or the bottom of the third all the way to the top of the fourth, you have to have these long interhelical string spaghetti things shown in pink. And that can destabilize the protein and not make it sort of a lean, mean version of IL-2, which is this Neo2 molecule that we're calling it, that has these nice short linkers and eliminates, right, all of that spaghetti. So we popped out a variety of different designs computationally, and then we used the yeast display platform that I mentioned earlier to display all of those different designs on different yeast. And then we came in with the beta gamma receptors and selected for things that were able to bind to that target. And through various iterations of this molecular design, right, sort of Darwin in a test tube, like we talked about before, we were able to come out with a molecule that had beautiful binding to the IL-2 or beta gamma receptors, but no interaction with IL-2 or alpha, in contrast with the wild type IL-2 cytokine. In addition, we saw that the IL-2 or alpha positive cells normally have the bias, right, for IL-2, but in this case, Neo2 was active on the effector cells, giving the overall advantage in terms of signaling to immune effector, right, and immunostimulatory responses. Finally, we demonstrated that this sort of super hyperstable cytokine that we had created was basically a rock, right, indestructible. We could actually heat this to 80 degrees Celsius for 2 hours, and it lost no activity. And we could even literally almost boil it to 95 degrees Celsius for hours at a time without any loss of function, whereas IL-2, right, the regular cytokine, was dead in the water. And more importantly, right, we had created something that was biased toward activating the immune response. And in contrast with IL-2, which was not able to control this very aggressive model of colorectal cancer, our Neo2 molecule increased survival and significantly inhibited tumor growth. The same thing happened when we used it in a melanoma tumor model. And actually half of the mice, right, in the melanoma model were sacrificed due to the toxicity of the drug. And the other 5 died due to the tumor burden when we treated with only the natural cytokine. But then when we came in with Neo2, right, our engineered computational cytokine, we had a 50% long-term survival rate. And I don't know about you, but I'd rather have my odds be 50% than 0%. So that's huge progress for a very aggressive model of melanoma. Finally, we didn't see any immune response, right? One of the problems that I mentioned about natural cytokines, or sorry, about natural proteins, is they can lead to immunogenicity, meaning an immune response against the exogenously delivered cytokine. And fortunately, we didn't see any evidence of that that was significant in this case. So I just want to wrap up there so that we can leave a few minutes for you guys to ask all of your burning questions about protein engineering. But I just want to say that, you know, I've had such a positive experience in the protein engineering sphere, but also in the scientific sphere, just with people like you who are here today, who are just curious to understand nature better and how we can actually manipulate, right, what's in nature to create the next set generation of therapeutics that will transform human health. So, and of course, acknowledging all of the awesome folks in my lab at the Translational Tissue Engineering Center at Johns Hopkins School of Medicine, and acknowledging you guys for your attention and for coming here today. And please let me know if you have any questions.
Thank you so much, Dr. Spangler. That was an amazing talk. I have one question. It's like more broad, but with like biomedical engineering, I know like when I first learned about the field, they said it was basically like a combination of CS, physics, and biology, and chemistry. Which of those 4 subjects do you use the most with biomedical engineering?
It's a great question. And I would expand that. It's definitely biology, physics, chemistry, all of those things are in there. Mechanical engineering, material science and engineering, right? It's really, it's a lot of different things because, and I think when people ask, you know, to sort of define, you know, what is bioengineering, what is biomedical engineering, what it really, I think at its core, right, is using quantitative tools and technologies to affect human health, right, in some way, shape, or form. In my lab specifically, on a daily, you know, sort of basis, we use a lot of chemistry, a lot of biology, specifically immunology, and a lot of physical engineering principles, I would say, are the primary things that we use. But you could look at another biomedical engineering lab, and it could be a completely different subset. They could use a lot more computational tools. They could use, you know, basically just be straight coding all day, like, you know, no wet or experimental, you know, sort of component. There are other labs that are very biomaterials focused, right? And they do a lot of material science and chemistry, and also, you know, biological, you know, sort of experimentation as well. So I guess I sort of artfully dodged, in some senses, committing to an answer in your question. I can definitely speak for my lab and I can speak for, you know, specific labs, but the exciting part about the bioengineering field, which is what really drew me in the first place, is that you're constantly exposed to many different subjects and many different branches of science, and you don't have to necessarily define yourself or commit yourself to just one field.
Thank you. That does answer my question in many ways. But have you personally like used computer science?
Yeah, so great question. So my lab collaborates with David Baker's lab and then also with Jeff Gray, who's in the chemical and biomolecular engineering department here at Johns Hopkins, for most of the computational components. And so we don't actually do the coding, you know, sort of interface with Rosetta, but we get the outputs of that. And we visualize. I showed you, you know, some different protein structural diagrams, you know, how we would visualize, and we interpret that by using these computational algorithms to determine what interactions are happening, what non-covalent, right, what van der Waals, hydrogen bonds, right, what's happening between the different amino acids in the interface. And we synthesize that information into how we're going to design the libraries. So although we're not the ones necessarily developing the next generations of software, we're using those softwares to integrate into the engineering process and the design process for our work.
Thank you.
So it looks like there's some questions from the chat here. Which specific components of the protein did you engineer to make those proteins that can survive high temperatures? Great question. So what we did was we actually packed the core of that fourth helix. Let me see if I can go back to the design here. So you can see that this helix that's shown in green is kind of bent, right? It has sort of like an L shape to it. And we flattened that out. You can see here shown in yellow. So we changed the order of the helices a little bit, which is fine. But more importantly, right, we flattened out that helix in the back so that essentially, instead of being, sort of the helix that would kind of recruit in that interface with IL-2 or alpha, now it's just packing flat and tightly against the other three helices to allow for the protein intramolecular interactions, right? The interactions holding the protein together to be more stable. And that stability is what led to ultimately having the thermal stability, because even if you heat this to very high temperatures, those alpha helices, right, are very close to each other and they're optimally packed so that those interactions will be very, very hard to destroy, even though they're non-covalent. The other thing that we did, which I mentioned before, sort of with the spaghetti analogy, is that instead of having these long interhelical loops, right? So the most flexible parts of proteins are what will destabilize them the most, right? Because that stuff is dynamic and it's floating around in space rather than having a very regular and regimented structure held in place by hydrogen bonds, which occurs in alpha helices. So we basically eliminated everything but the alpha helices, right, by creating these very short linkers between the helices. And now there's no unstable parts of the protein that can lead to it falling apart. And as a result, it takes very, very, very high temperatures that we weren't able to reach in these studies to be able to destroy our engineered cytokine. Great questions. Other questions from the audience? Or, how do you flatten the helices? Great question. So interestingly, the sequence between IL-2 and NEO2, if you aligned the sequences, it would be 100% clashing. There's no resemblance between the sequences at all. And the reason for that is we just rebuilt the entire helix piece by piece. So we took these sort of four, like a sliding scale of 4 amino acids, and we said, how will these 4 amino acids look, right, in theory? Because it would be very hard to just take 100 amino acids and say, how is this thing going to fold and how is it going to look? Instead, we looked at different pieces and we mapped 4 amino acids at a time. And for each of those 4, we optimized them to be in this alpha helical conformation. And so again, when we had sort of this bend in the helix, it had a region that was not going to stably hydrogen bond with the adjacent residues, and that's what led to sort of it bending. And in ours, we designed it so that there would be no gaps and no spaces in between the hydrogen bonding network that holds together that alpha helix. Again, great question. Thank you, Julia. Other people. Oh, Julia has another question, which is great. The function of the protein, was it different when you engineered it? Indeed, the NEO2 function is different from IL-2, and the biggest difference is what you see here between cells that express IL-2 or alpha versus the ones that don't. Because remember, we designed this NEO2 so that it no longer cares about alpha, right? There's no interactions, it's just a total flat line. And as a result, we got a bias toward immune effector cells that ultimately led to its efficacy in treating various types of cancer. And we're also experimenting with the same molecule in infectious disease models. Because again, the same idea and concept of activating the immune system can be used for cancer or for various infections that would threaten the immune response. Why is thermal stability important? Sean asks. Yes, it's actually a very insightful question that you ask, and one that we put a lot of thought into in terms of how we were going to frame this project and ultimately the paper, that came out of this work. And in some sense, we've explored, what are the advantages of having a thermostable protein. And in some senses, there's much that remains to be explored. So one advantage, of course, is that potentially at under high temperature or pressure conditions or situations, this protein could be extremely stable. So you think about drug manufacturing and drug processing, having a molecule that you can produce at very high levels and not have to worry about the storage conditions could be extremely useful for disseminating it all over the world, right? To different regions in different places, and having that be able to withstand the elements and other sorts of challenges that might arise. It can definitely help cut down costs in terms of manufacturability and potentially facilitate FDA approval and advancement and ultimately clinical application. Other things from a research standpoint are if you actually have a protein, right, that can take really, really high temperatures. There's all sorts of processing and development that you can do in the research lab context. Another example would be to actually use this in what's called hyperthermia conditions, where there are some approaches now, particularly for cancer therapy, where they try to use heat treatment or heat shock to destroy cancer cells. But when you're using this heat treatment, if you're giving, another therapy in tandem, you could destroy that protein, right, by elevating the temperature. But this protein, right, would be very robust against any temperature increase that you would use for any sort of hyperthermia treatment. So there's a variety of different applications, both tapped and untapped, that we're still thinking about. But at the moment, we're just really excited that we've created this molecule that seems to be behaving exactly as designed, right? In sort of this bespoke, kind of condition, but also that it could have, larger implications than we can even envision right now.
Thank you, Dr. Spangler. I think we have, sorry to cut you off, but we need to move on to the next speaker.
Of course. Yeah, thank you again for the opportunity. And if anybody has any questions, I had my email address up here at the end. Please feel free to reach out anytime. And also to follow up in the chat if you have more questions.
Yep. Yeah, thank you again for the amazing lecture.